A very warm welcome to this UNICA student webinar on clinical drug development in the COVID and post-COVID era, organized by UNICA in cooperation with the Pharmaceutical Student Association and the European Medicine Agency. I'm Luciano Saso, I'm the president of UNICA, and I take this opportunity just to mention the organization of UNICA, which is an association of, at the moment, 53 universities in 37 capital cities. And it is a pleasure to organize activities like this one for students of our network and discussing such an important topic in this period of developing drugs. And as you know, of course, vaccines are important drugs that has been developed recently. And we already started this series of webinars, for students with other topics, and we will continue. I mean, we received so far a very good response from UNICA universities and beyond, and we think that this is something that will be very useful for you. This webinar will be recorded. Also, we are going to send a survey to all of you to see your, level of satisfaction. We hope that all of you will like this kind of initiative. Please send us also, feedback if you wish in the future to improve this kind of initiatives. I want to thank very much all the participants of these webinars. First of all, as I said, the partnership with the European Pharmaceutical Student Association. We will have in a few moments a few words from Maeva D'Almeida, who is a Vice President of Internal Affairs of this organization. I want to thank very much the speakers of these initiatives, and in particular Professor Josse Thomas, who will give a talk about novel clinical drug designs, and is a guest lecturer at KU Leuven in Belgium and senior consultant of the PharmaCS, and Professor Hans-Georg Eichler, who is a senior medical officer at the European Medicine Agency. And he will give a talk on drug development in the COVID and post-COVID eras, the perspective of regulators. And also I want to thank my friends and colleagues, Professor Schravendijk, who is Emeritus Professor of the Faculty of Medicine and Pharmacy of Vrije University of Brussels, and Professor Sibel Suzen from the Faculty of Pharmacy of the University of Ankara, who will animate the discussion after the two talks. I invite all participants to ask questions in the chat at any moment. You can do that, and then we will actually address your questions after the two presentations. At the end, we will have the discussion. So without further ado, I just leave the floor to Maeva D'Almeida also for her welcome. Thank you.
Thank you, Luciano. Hello, everyone. My name is Maeva D'Almeida. I'm the current Vice President of Internal Affairs of EPSA, which is the European Pharmaceutical Student Association. We represent over 100,000 pharmacy students around Europe from 35 countries. And our mission is to actively engage students and professionals trying to bring together pharmacy knowledge and those students together. We would like to thank UNICA for providing us with this opportunity to co-host this webinar on the drug development, the clinical drug development in this COVID and post-COVID era. This is a highly interesting and important topic for us as future pharmaceutical professionals, and we are very looking forward to all these discussions. To the students, feel free to ask any question, to really take this opportunity with these high-level speakers to get more about this topic, and really we hope that you will get fruitful discussions and good knowledge at the end of this session. Thank you very much.
Thank you, Maeva. And now it is a pleasure to leave the floor to Professor Josse Thomas, who will give a talk on novel clinical trial designs. please.
Okay, can you see my slides?
Yes, perfect, thanks.
Okay. So I will start with the outline. After a brief introduction, I will discuss adaptive designs, which are the basis of a lot of the novel clinical trial designs, and then move on to more complex innovative designs. That is the terminology used by the FDA. It is also known, or they are also known as complex clinical trials, the terminology used by the EMA, or master protocols, and they include basket and umbrella trials, platform trials, and multi-arm, multi-stage trials, so-called MAMS. Then I also briefly touch upon seamless designs as they are used in seamless clinical drug development. Finally, a few words about other recent trends and a conclusion. Why are these novel designs being introduced? Because traditional and conventional clinical designs have been challenged. They are studying one, maximum 2 drugs, let's say, in one disease with a fixed design in general, either parallel group or crossover, and they are time-consuming, deemed to be expensive, less efficient, and not adapted to new trends, and in especially the emergence of personalized medicines. Novel designs, and then especially the novel designs with adaptive features, introduce more flexibility to cope with continuously emerging new knowledge, and they are able to address multiple questions and also sometimes in multiple subpopulations in one trial. They increase the efficiency of clinical trials and the success rate of drug development by quicker detecting target subpopulations, earlier detection also of drug failures, and so less waste on unsuccessful drugs. Let's start with adaptive designs, their main features. The definition is that they use in-trial accumulating data to redesign the study without undermining its validity and integrity. And they're redesigning the study when the study is still running. Possible adaptations include, as examples, a change in sample size, a change in the randomization, or the dose escalation, dropping or adding treatment arms, or one treatment arm, dropping subpopulations, the so-called losers, enrich subpopulations, the so-called winners, and then also to stop the trial early, either for efficacy, safety, or futility. They are a special type of so-called flexible designs, which is a more general term, but the specificity is that these flexible designs, these adaptive designs, are all the modifications should be pre-planned or pre-specified or predefined in the study protocol. And that is the main specificity within the group of flexible designs. There are many types of adaptive designs, each with specific advantages and disadvantages. Some exist already for decades and others are more recent. A number of possibilities here on the list: adaptive randomization designs, group sequential designs, sample size re-estimation designs, and I will come back to all these, drop the loser design, adaptive dose finding designs, adaptive hypothesis designs, so you can adapt, let's say, everything within the clinical trial methodology. You can adapt also the endpoints if they are pre-specified, if these adaptations are pre-specified in the study protocol from the beginning. We also know seamless adaptive designs. I will also give an example of those. And then combinations of these different design types in what are called multiple adaptive designs. So they come in many shapes and sizes. I will now, this is a schematic of the adaptive designs within the flexible designs. The flexible designs, the flexibility in those designs is unplanned. And so the studies evolve without or with unknown properties beforehand. Adaptive designs are a type of flexible designs where everything is pre-planned in the study protocol as much as possible. In the learning phase of the drug development, the clinical drug development, examples of adaptive designs are adaptive dose response designs or adaptive dose response either for safety or for efficacy. And then in the confirmatory phase, most what we see is adaptive randomization designs, group sequential designs, sample size re-estimation designs, and the combinations thereof. And then in between you see combined seamless phase designs where the phases of the clinical development are combined in one seamless trial, either from phase 1 to phase 2A, for instance, in early clinical development, or from phase 2B to phase 3 in the late clinical development. Let's start with a few more details now on the different types of randomized, adaptive designs. First, the adaptive randomization designs. The traditional randomization fixes constant allocations in advance without changing them during the course of the trial. For instance, a 1-1 randomization between the drug and placebo, or a 2-2-1 between different arms, the experimental drug, reference drug, and placebo, for instance. In adaptive randomization, you can change the allocation of subjects to treatment groups based on information that you gather from the trial, from intermediate results within the trial. And that is to increase the probability of success of the trial. There are essentially two types of randomization, of adaptive randomizations that are used. Either covariate adaptive randomization, then you allow to change the randomization due to the distribution of covariates already in subjects already included in the trial. And then the best example of that is minimization, and which is a form of adaptive stratified randomization. Another type is response adaptive randomization, which is also much used. And then you observe a number of treatment outcomes intermediately, and then you adapt the randomization according to the response that you have seen to favor the better treatments and the better treatment arms. It maximizes the power and minimizes the expected failure rates. Second type of adaptive designs is group sequential designs, which are already there for a number of years. That is a type of design in which the only interim decision is whether to stop the trial early for harm, efficacy, or futility, the whole trial or part of the trial, after one or several interim analyses. The sample size is not fixed, and the accumulating data are periodically analyzed with the intention to stop the trial when a pre-specified stopping criterion is met. So again, the stopping criteria should be pre-specified in the study protocol from the beginning. The interim analyses are best done by an independent data monitoring committee and an independent biostatistician. They are used already for several decades, as I said, especially in oncology. And the most familiar example is the 3+3 phase 1 design that is still widely used in the development of anti-cancer drugs. Another example also much used in oncology is this 2-stage design by Guillen, Simon, or Pocock. These are typical examples for phase 2 designs in oncology. And a more recent example is a more complex one, is the so-called multi-arm, multi-stage design one, and I will come back to that one. These are typical examples of group sequential designs. A schematic of such a group sequential design, you have your population, your study population divided in two arms, for instance, by randomization, one on the top to be treated with one drug, and the second one on the bottom to be treated with a combination of two drugs, for instance. The first interim analysis after a certain period of time would be on an intermediate endpoint, the progression-free survival, for instance, and a second interim analysis could be done later on or at the end of the trial on overall survival. And that is typically a schematic of a group sequential design. Another type of adaptive design is the so-called drop the loser design, also known as the pick the winner. That's the alternative, or also called enrichment designs when we work with, for instance, genetic typing on biomarkers. It is a two-stage or even a multiple-stage adaptive design that allows dropping inferior treatment arms while keeping the control arm and keeping also, of course, the more successful arms. Particularly useful when there are uncertainties about the dose levels in the transition from phase 2 to phase 3 clinical development, for instance. They are often combined with other types of adaptive designs or adaptive features, response adaptive randomization, thus increasing the probability of participants being randomized to groups with the best chance of success. An example in the next few slides, also one with a biomarker adaptive enrichment design. This is a drop the loser adaptive design. You start the trial with 4 arms, for instance, 3 active ones with 3 different doses, for instance, and a placebo group or a standard of care treatment group. In this case, a placebo group. Then you observe the predicted response and you adapt the randomization in the different arms according to the response that you see, to the intermediate analysis response. And then, for instance, in the first dose, the lowest one, for instance, that arm is dropped. The second one is continued. The third one is also continued for a while. And then the next one is dropped again. And finally, you drop the inferior arms, all the inferior arms, and you adjust the sample size and you just continue with the second one, for instance, the dose that is apparently the most successful one versus placebo in the confirmatory phase. This is typically a design called the drop the loser or pick the winner design. The next one is an enrichment design based on biomarker responsiveness. You start the trial with a mixture of a patient population that is either biomarker positive, the darker ones, or biomarker negative. You screen them before randomization, and then during interim analysis, you see, for instance, the first result, which is response in the biomarker positive ones and response in the biomarker negative ones. Then you just continue the recruitment without any restriction. The second possibility is after an interim analysis is that you see response in the biomarker positive population, but no response in the biomarker negative population. And then the adaptation is that you restrict the recruitment to biomarker positive patients and you drop the biomarker negative ones. Third possibility is that you have no response neither in the biomarker positive patients nor in the biomarker negative ones, and then you stop recruitment and the trial stops for futility because you will not be able to demonstrate any positive or negative result at the end. In general, what are the pros and cons, the advantages and disadvantages of these types of adaptive designs? They increase flexibility, that's right. But as I was saying, the specificity of adaptive designs versus flexible designs in general is that all the adaptations should be pre-specified in the study protocol. There is no need to file as many protocol amendments. Of course, if you have complex trials, and I will come back to that, or I will come to that, then you have still a number of amendments, but we'll see why that is somewhat different. You have earlier detection of drug failures, more efficient use of trial participants, because you only keep the ones with possibility of more success. You have overall savings in time and costs. But the challenges are that they are not always fully accepted by regulatory authorities, although there has been a lot of evolution into that, because what is also very important, that you should keep the validity, the scientific validity and the scientific integrity of the trial. There is a lot of acceptance already within regulatory authorities in the US and in Europe, but to make it as drug development is worldwide, to make it internationally fully accepted, there is still a way to go. Another challenge is the shifting of the target population. Of course, you start with the target population, but you can end up at the end of the trial with a different patient population. And how are you going to deal with that? So they are logistically complex. That's a challenge or a disadvantage eventually. And then there is not always sufficient expertise in-house within the company, within the CROs that help pharmaceutical companies and sponsors, within the regulatory authorities, within the ethics committees, etc., etc. You also need independent governance, because these trials are complex, need complex and novel statistical analyses, and it is interesting to have then independent data monitoring committees, independent steering committees, and independent statisticians, especially when they become more complex. And that is where I'm coming now to what is called complex innovative designs, or complex clinical trials, or master protocols. They are based on these innovative designs. They use extensive features of innovative and adaptive designs. The common features of these complex innovative designs, or complex trials, or master protocols, are that they use one overarching protocol to answer multiple, or to address at least multiple questions, evaluating multiple interventions in one single disease, or a single therapy in multiple diseases or multiple disease subtypes and subpopulations. There is an overarching master protocol and specific what are called sub-protocols for the different parts of the study and the different treatment arms. Multiple and complex adaptive features are present and extensively used. And for instance, you can add or stop interventions, and you can add or stop subpopulations. Another common feature is that they use, most of them, used a common screening platform and a common operational, a complex operational framework. Well-known examples of these complex innovative trial designs are basket trials, where a single therapy is studied in multiple diseases, umbrella trials, where multiple therapies are studied in single diseases, and then what are called platform trials, which are in fact perpetual or so-called standing umbrella trials, where therapies are allowed, or interventions in general are allowed to enter or leave the platform. That's what they are called, the study, or leave the study, the platform. That's why they are called a platform trial. You can get up on the platform or you can get off the platform. They're also known as multi-arm, multi-stage designs, MEMS designs. And I will now come into somewhat more detail about these trials. The first ones are umbrella and basket trials. They have been introduced with the ascent of precision medicines. They are widely used in oncology. The umbrella trial in oncology studies one type of cancer with different genetic modifications. There with the colored dots. Lung cancer disease, for instance, with different modifications, genetic mutations in different patients. And in the one mutation, you study drug 1. In another mutation, you study drug 2. And in a third mutation subset, you study drug 3, all within patients with, for instance, non-small cell lung cancer. The second type of trial is the basket trial, where you do exactly the reverse. You study multiple types of cancer, in this schematic, lung cancer, pancreatic cancer, brain cancer, and colon cancer, with one common genetic mutation. The common genetic mutation is the same in the different trial, in the different cancer types, and you test one drug that targets that specific mutation. I will go through 3 schematics about these 2 types of trial. This is schematic about conventional oncology trial, either in sequence or in parallel. On the left side, you see different types of tumors, either the squares, the circles, the diamonds, or the triangles, tumor A, B, C, and D. And then the colors have indicate different types of mutations, 1, 2, 3, and 4. In a trial population that you see here with different types of tumors and different types of mutations, classically what you do in conventional oncology trials is that you do the trial per type of cancer. Trial 1 with drug 1 in, for instance, lung cancer on the top. Trial 2 with drug 2 in, for instance, pancreas cancer. Trial 3 with drug 3 in, for instance, breast cancer. And trial 4 with drug 4 in another type of cancer. Now, in a basket trial with the same amount of the same target or study population, what you do is you subdivide them in patients with different types of tumors. On the top, you see 3 different types of tumors. On the basis, you see 2 different types of tumors. And then you do that, the different types of tumors and organs. You see that on the top, they all have the same mutation. And you test in the different types of tumors with the same mutation, one drug. You can also do that in another trial with another drug for another type of mutation. In an umbrella trial with the same set of study population, you see now on the top a trial where you have different mutations in one type of tumor. The tumor type is the same, but you use different drugs in different subsets of genetic mutations within these tumors in different subsets of patients. Trial on the top, trial on the bottom. This is a schematic of an umbrella trial. A multi-arm, multi-stage MEMS design. This is a schematic of how that looks like. Here you have different regimens of drugs, either drugs alone in different doses or a combination of drugs versus a control regimen, and that is often the standard of care and the current standard of care. At the first interim analysis, for instance, you drop the second arm. The second interim analysis, you drop 2 other arms, and then you continue with the most successful ones versus the control regimen. This is what is called a multi-arm, multi-stage MEMS design. Also with different features of adaptive designs as the group sequential design, for instance. This is a schematic from the New England Journal, which is a bit more explicative. Potential design of a platform trial involving a single disease. This is a type of a platform trial or a MEMS trial. What they often use is a common screening platform. You see on the left that you can do screening for 2 biomarkers, biomarker A and biomarker B. And either the subpopulation is biomarker A positive, and those patients will go on in the top of the schematic. Either they are biomarker B positive, and that is the middle stratum, or either they are biomarker negative, and that is the lower stratum. In the biomarker positive stratum, you can investigate drug 1 and drug 2 versus the standard of care. In the biomarker B positive, you can use another drug, investigational drug 3, versus another type of standard of care in biomarker negative or in biomarker B positive patients, sorry. And in all the biomarker negative ones, you can use another drug, investigational drug 4, against the standard of care for biomarker negative patients. A bit further on the top, you see that after a first interim analysis, you stop the investigational drug 1 because criteria for success are met. Then a bit later on another time point in the time as the study is ongoing, you can introduce a new drug in those biomarker positive patients, investigational drug 5, for instance. After a while in that same stratum, because you stop investigational drug 1 because of success, that investigational drug 1 can now become the standard of care. That will be the new standard of care in that stratum. The same time here in this biomarker negative stratum, you stop for futility because apparently investigational drug is not that active and you will not be able to show any success or failure at the end of the trial. These patients are then going on in the biomarker negative striatum, and then they will continue in the trial in the lower part of the ones that started from the beginning. Because now you have, you can also introduce another biomarker, biomarker C, for instance, because biomarker B was not that interesting apparently, because you had to stop that stratum for futility. And now patients are randomized between biomarker positive ones and biomarker A positive ones and biomarker C positive ones, and then the biomarker negative ones, again versus their own standard of care. So this is a bit more complex already, but this is a good schematic to give you an idea of what are the different possibilities. This is just for your safety, for the sake of being total in everything, so that you have the capture with that goes with the figure. Now the EMA has set a number of criteria for the clinical trial authorization submission. Either these trials, which are complex, are introduced to them as a CTA as one trial, and then you introduce the master protocol, which sets out all the common features and what will be adapted in the future, together with the first sub-protocol, which details which type of drug, which type of biomarkers and everything will be done in that part of the study. And then as the study goes on, you introduce sub-protocol 2, sub-protocol 3, etc. as the study goes on. That is one possibility. You do that as one trial. You introduce the master protocol from the beginning with one sub-protocol, and then with substantial amendments, you add sub-protocol 2, etc. Another possibility, as you consider them as separate trials, and then you introduce the master protocol with trial 1 as one trial. You get a separate UDRA-CT number. You introduce the trial number 2 as a separate trial with a separate UDRA-CT number, and you also introduce the master protocol that goes with it. And you can continue like that. If you want to introduce a third trial, separate one, you introduce the protocol, get a new UDRA-CT number, and you also add the master protocol with that introduction. These are two modalities of CTAs that the EMA has set forward in their guidance for complex clinical trials. 3 examples of complex trials now in cancer. This is a basket trial. I'm not going to go into much detail. It's an early phase 2 trial with a Simon's 2-stage design in non-melanoma cancers with a BRAF mutation. The next one is the Battle 1 trial. It's an umbrella trial with different targeted therapies in chemotherapy refractory non-small cell lung cancer. It's also a phase 2 trial with adaptive randomization design. And then an example of a MEMS trial or a platform trial, the Stampede trial, which is one of the best known and one of the earliest ones of these type of trials. And it is still ongoing. It started in 2005, and there are more than over 9,000 patients who joined the trial up to now. It is the best. The question is, what is the best way of treating men with advanced prostate cancer? And the results so far demonstrate that adding docetaxel or abiraterone improves disease control and life expectancy. You can find that all the details on the Stampede website. And it is now open for 2 new research questions. And that is, can adding metformin in non-diabetic patients improve life expectancy? And can hormone injections be substituted for hormone patches? And I have 2 schematics about the Stampede trial here that you can look at more closely in detail. I will quickly go over it. Patients eligible for Stampede with prostate cancer and decided to have planned radiotherapy and docetaxel. And either in the left panel, they have a maximum of 4 weeks of luteinizing hormone, release hormone injection for their treatment, for their cancer treatment, or all the other patients on the right side. And on the left, there is sub-randomization of non-diabetic patients and diabetic patients. On the right side, the same. And then you have different treatment combinations that are available to these 4 subsets of patients. Now, in the meantime, there are already more than 4 of these subsets and more than, I think, 7 or 8 different treatments that have been tested or are still in test mode. The standard of treatment is a combination of radiotherapy and androgen deprivation therapy. Then there are different types of drugs. You see, started in 2005, 2006, still running. And then the last 2 that were at the arms that were added, metformin or the transdermal estrogen therapy, they are still running today. Master protocols were introduced and these complex trials were introduced in oncology, but now you see also examples beyond cancer. And then a lot of them also in infectious diseases. And here are a number of examples, just to cite you a few examples. You can look them up and have a more detailed look on the websites of the different trials. The ADAPT trial, which is a platform trial of antibiotic drugs targeted for resistant pathogens. The PREVAIL-2 trial was stopped, was a platform trial of multiple therapies in Ebola virus disease. It was stopped because the Ebola epidemic stopped in Liberia, so the study could not recruit anymore, and there are no results available. The Panacea TB trial in anti-tuberculosis antibiotics, and also recently, since the COVID-19 pandemic, several trials initiated in COVID-19 patients. Mostly hospitalized patients for the moment, but also now there are already 1 or 2 trials in ambulatory settings. Different examples here: the Solidarity trial initiated by the WHO, the Recovery trial by Oxford University, and the ACTT trial initiated by the NIH. I will come back to it immediately, but I will go on first with other disciplines and other therapeutic areas where these types of trials have been introduced more recently. The DIAN-TU or TU001 trial is a platform trial of disease-modifying treatments in patients with Alzheimer's disease, a special type of Alzheimer's disease. In intensive care, the PREPARE study, the PRACTICE study, Study C, different platform trials of 4 treatments in patients with severe acute respiratory infections. And there are many more to come, certainly in different sizes and shapes. And here are 3 examples within the COVID pandemic. The Solidarity trial of WHO, which is a platform trial, in hospitalized patients. The arms that are stopped already have either a positive outcome, which is the arm of dexamethasone, which is now considered as a treatment for hospitalized patients or subset of hospitalized patients that are severely that have severe disease. And there are also arms which have been stopped because of lack of success. The hydroxychloroquine arm, the lopinavir combination with rotinavir, which are already known antivirals for other diseases. Interferon beta has been stopped, and remdesivir, which also a antiviral that has been used in Ebola, that's been tested in Ebola, was not successful there neither. And there are a number of other antiviral drugs, immunomodulators, and the anti-coronavirus 2 monoclonal antibodies that are considered for evaluation. And also Solidarity is considering to have a special subset of trials of platform trial for vaccines. So the former ones are called Solidarity Therapeutics, and the new one on vaccines will be called Solidarity Vaccines. Another platform trial in hospitalized patients is the one from University of Oxford in the UK, which called the Recovery trial, randomized evaluation of COVID-19 therapies. Again, a number of arms have already been stopped, or at least one for a positive outcome, the dexamethasone one, and then 2 for a negative outcome, the hydroxychloroquine and the lopinavir rotinavir combination. There are still 3 arms currently recruiting. One arm which is studying tocilizumab, which is an anti-inflammatory agent important in the hyperactivity reaction that you see in a lot of these patients. Convalescent plasma with antibodies from COVID donors, of plasma donors of patients that went through the COVID disease already. And then the monoclonal antibody cocktail from Regeneron. These are in the recovery trial arms that are currently recruiting. I'll go through some advantages now and challenges within these protocols, within these complex trials. The advantages are clearly that they have built-in flexibility, predefined, pre-specified flexibility, adaptations, and that they are highly dynamic in design. Another advantage is that they use, most of them, used common screening platforms for biomarkers, which allows then to test subsets of populations, which is interesting in the era today of precision medicine. There is shared use of control patients. They have shared standard of care arm, and that also allows that the standard of care evolves over time, that the standard of care can be changed over time. It is an advantage also that you can use shared governance, centralized shared governance. The IDMC and the independent steering committee can be used for different types of drugs, different types of subsets of diseases, and also with different types of companies, because you can also experience or study drugs from different companies so that they have to collaborate. And that is maybe an advantage, but also a challenge. Common data management and statistics, I already alluded to that too. So you gain in efficiency, you gain time, you gain resources, and finally also, why not gain in overall quality, as long as the scientific integrity and data integrity is maintained. The challenges is that you have to work a lot more upfront, because everything, well, the biggest possibilities have already to be put in the protocol from the beginning. So you have increased upfront planning and coordination, which is essential. The designs are more complex, and of course, during the study, you need to do more and more complex real-time decisions. They also need novel and more complex statistics because of the multiplicity problems that you can imagine. And so you need extra experts to do that. And mostly it would be better to have these statisticians independent of the sponsors and of the sponsors. You also have changes in the marketplace, as these trials can last very long, many years, as you see with the Stampede trial, already there for 15 years. And the market changes. There are introduction of new drugs. You have to cope with all that. And that makes it complexer and more difficult. And then there is also the challenge of the universal acceptance by regulators. So in summary, there are operational, statistical, and regulatory hurdles. Now I'll move on to another type of adaptive designs, which are called seamless adaptive designs. I have put them at the end because they are used in what is called now seamless clinical drug development. Seamless adaptive designs are designs that combine the aims of separate phase 1 and phase 2A trials in early development, or phase 2B and 3 trials in late development. And you combine these trials in one single trial with a lot of adaptive features. Such trials are conducted in 2 stages at least, or sometimes in multiple stages, each separated by an interim analysis, so they are by definition group sequential designs, but not by a long pause. And so they save a lot of time in the development of new drugs. There are 2 types of seamless designs possible. What is called operational seamless designs. You just seek to exploit the saving in time. But the more complicated ones are so-called inferentially seamless designs, where you use statistical methods that combine the data from the 2 phases. For instance, from the phase 1 and from the phase 2A trials, which are classically or conventionally done separately. Here you can combine these data from phase 1 and phase 2, for instance, or for the 2 different phases in seamless adaptive designs. This is an example, a schematic of such an adaptive seamless design. On top, you have seen these, of course, already, but now this is a phase 2 trial combined with a phase 3 trial. The first part here is a phase 2 trial where you see 3 different doses, for instance, of a drug versus placebo, and you drop at intermediate interim analysis already the first one, you drop B, the second dose. A bit later in the interim analysis, you drop A, the first dose, and you continue the trial by increasing the number of participants, by increasing the sample size, pre-planned in the design already. And that is the phase 3 part of the trial. And then you see that you can condensate in time the time needed to get to the end of phase 3. This is a schematic below here of the classic phase 2 trial, and then afterwards followed up by a phase 3 trial. Here you do the classic phase 2 trial, and you do data analysis. You have the results available. You start planning the phase 3 trial, and then you do the phase 3 trial with the chosen dose C, for instance, versus placebo, and you end your phase 3 trial a lot later. So that is an interesting evolution, seamlessly going from one phase in the clinical development to the next one by using seamless designs. The ultimate evolution is what is called now seamless clinical drug development. The first example of such a seamless clinical drug development was the development of pembrolizumab, Keytruda from MSD, which is an immune checkpoint inhibitor used as an immunotherapy in cancer. In 2011, the first in-human trial was started, which had impressive responses and durations, especially in melanoma and non-small cell lung cancer. Then that trial was given, or the drug, was given in a year later by the FDA breakthrough therapy designation, which makes it the possibility of developing the drug faster. It was with an increase, the sample size of the first in-human trial was increased to 1,200 patients with multiple expansion cohorts, evaluating different dose regimens and different predictive or candidate predictive biomarkers in what is called now a supersized phase 1 trial. In 2014, they got accelerated approval in melanoma with 173 patients, and later on approval in non-small cell cancer and a companion diagnostic. And today you see numerous examples. Go and have a look at clinicaltrials.gov, and you see numerous examples of drugs in development with trials of seamless designs and seamless clinical drug development strategies. I will briefly touch upon a few other recent trends in clinical trial methodology, except of these novel designs that I spoke about. just to mention them, I'm not going into detail. You see more and more extensive use of clinical trial modeling and simulation. The introduction of pharmacometrics dates already back for a number of years, but now we also speak more and more about model-informed drug development. You have already seen model-informed drug design, but now more and more it is introduced in clinical trial world with model-informed drug development, modeling and simulation. Another initiative is the innovative use of external data, eventually with Bayesian design features, historical data used in novel ways, register-based randomized clinical trials, which are randomized clinical trials which are piggybacked in epidemiology register, either of a disease, either of a drug. And then also because we see more and more conditional approvals, and then these have to be followed up, the marketing authorizations have to be followed up post-marketing authorizations. You see also number of different types of trial designs into the real world after marketing authorization. An example here is mixed treatment comparisons, where trials have been done placebo versus one drug, and then placebo versus another drug in the same indication. And these are possibilities to mix now the treatment comparisons if they have several endpoints in common and things like that. But that asks for thinking about when you design a trial, think already about possibilities of mixing them with other trials that in the same indication later on. Also, what is called now prospective individual patient data meta-analyses. I've seen lately one of these examples for convalescent plasma treatment in COVID-19 patients. There, prospectively, when the platform trial for convalescent plasma was introduced in COVID-19 patients, the investigators also thought already of combining the different treatments, the different studies with convalescent plasma all over the world by using individual patient data in a meta-analysis. When you think of that prospectively beforehand, you can characterize the study endpoints, you can harmonize these study endpoints over the different types of trials so that your individual patient data meta-analyses are much more robust. So this is what I wanted to tell you about. I want to conclude in the following way. There are certainly lots of opportunities with these types of novel designs. I've cited a few of them: increased flexibility and that are adapted to the new trends. For instance, the ascent of personalized medicines, which was the first trigger in oncology for introducing these type of trials. More collaborative efforts and more multi-sponsor trials. It is an opportunity to collaborate more in the COVID-19 period pandemic. We've seen that, and in fact, when the pressure is there, it seems to be doable, it seems to be possible. Multi-sponsor trials, collaborative efforts. With that, you gain in efficiency, you gain time, you gain resources, you gain quality. And that allows faster clinical drug development and ultimately earlier access to innovative drugs. The challenges can be summarized as the operational complexity and the operational feasibility. It is not always easy to do these type of complex designs that have been thought of sometimes by people at their desk. In the clinic, it is sometimes a lot more challenging, and also for the logistics, it's also a lot more challenging. The essential point there is, okay, with some operational complexity and complexity in the design, but the foremost thing is to maintain scientific integrity and also patient safety. It cannot go at the expense of scientific integrity and patient safety. Another challenge is the universal acceptance by regulatory authorities. Universal, I mean worldwide, and there is certainly a need for harmonized international guidance. And with that, because it is novel, it is still evolving, another challenge is the availability of experts and the training of experts in these complex trials. And that is investigators, biostatisticians, but also evaluators at the agencies, at the ethics committees, at the HTA bodies, etc. So experts are needed at the different stakeholder levels, and so there is a need also for training. And I hope I have added a small element to this training aspect. How does the future look like? Well, innovations, this is not the end, this is the beginning or the continuation of innovations that we will see in clinical trial methodology. They will continue to evolve, of course. They were already extensively used during the COVID-19 period, pandemic, and as they are here to stay in the post-COVID era. That is what I wanted to say about these trials, about the methodology of these novel trials. Thank you very much for your attention.
Thank you very much, Josse, for this comprehensive and extremely interesting presentation. I'm sure that all participants appreciated it very much. And now it is a pleasure to give the floor to Professor Hans-Georg Eichler, Senior Medical Officer of the European Medicine Agency. And he will give a talk on drug development in the COVID and post-COVID eras, the perspective of regulators. Please, Professor Eichler.
Thank you very much, Luciano. And thank you for inviting me to this interesting virtual meeting. I will now start sharing my screen so you can see, so you can see my slide deck. And at the same time, I will switch off my, if I can do that. No, I hope it's stable, the camera. I hope it is stable. If it's not, please let me know. So what Josse said is highly relevant, and I agree with everything he said, and I will revisit some of his topics, but I was also asked to briefly describe my organization, the European Medicines Agency, and also to elucidate how and what we do with academia, knowing that this is a university-based academic audience. So I will divide my talk into two very separate parts. The first is more on process, as I said, this is on us. And the second part will then be on science and COVID and non-COVID related development issues. So let me start with introducing you to the European Medicines Agency. This is an organization, an agency of the European Union. We are here to serve the 27 EU member states with around 450 million people. And it is probably underappreciated what we do. You would associate a regulator with someone who authorizes drugs for the market, but there is more to it. We want to be an enabling regulator, so we facilitate the development with a view to ultimately facilitating access to medicines for patients. Then, of course, we certainly evaluate applications. We are doing this now for the COVID vaccines, and I guess everybody knows that. But we also work once the drug is authorized, is on the market, and we monitor the safety, and I should say not just the safety, also the efficacy of medicines across their entire lifetime, and that includes the on-market phase. And lastly, we hope to provide reliable information on medicines in all European languages. Now, who are we? And I am keenly aware that this is not widely known, how a European organization like us operates. So we are essentially the hub for around 4,000 scientific experts from across Europe. And they collaborate in a number of scientific committees and working parties. And these are the ones who do the assessments and take the decisions. There is often a misconception that it is Brussels, or in our case, Amsterdam bureaucrats employed by the agencies who do take the decisions, who do make the assessments. Not the case, actually, or in our case, absolutely. This is very much in the hands of experts from the member states who convene in Amsterdam and then come to conclusions after they have made their assessments. Now, so this works as a medicines regulatory networks. We collaborate with the authorities that are based in the 27 member states. You may ask, why are there 50? Well, that is a trivial explanation. We do both veterinary and human products, and in many member states, you have different agencies to deal with those, and there are other anomalies, should I say. So it's about 50. Then it is the EMA itself, and of course the European Commission, which is sort of the legal executive arm of the regulatory function in Europe. The benefit of this for European citizens is that medicines are authorized in all EU countries at the same time. For industry, the benefit is there is one application, one assessment, and one marketing authorization for the whole of the EU. When it comes to safety, and I should add here efficacy monitoring, this is centralized. We coordinate that. And also the product information is available in all EU languages, and we take care of that. Now, how do we collaborate with academia? And I would say academia, that is people like yourselves, are a key stakeholder for us. We collaborate with individuals in different stages of their careers. We collaborate with academic groups, consortia, and also universities and learned societies. And, excuse me, this is a two-way street of communication. On the one hand, we get something, questions from academia. They come to us for training. They give us their expertise provided in the context of assessments, in the context of methods development. But on the other hand, we reach out to academia and ask them to consult. We want them to participate in workshops. They are present in our expert groups. We sometimes host academics at the EMA. The term SNE that you see at the bottom here means seconded national experts. So this could be someone who is an expert in this or that field based at the university of this or that town and who joins us, say, for a year to conduct work that could be research work at the EMA. And I think this is something you as young professionals may want to keep in mind and may explore this opportunity in your future. Now, what are we doing for academia and for innovation in the context of academia especially? We have a body called Innovation Task Force that is available to discuss with applicants various research questions informally. They can discuss any scientific, legal, or regulatory aspects of emerging therapies. Often this is with multidisciplinary groups, and we put those groups in contact with our working parties with our experts to enable the interaction between academics and regulators and regulatory experts. So that has been quite successful, and we I like to believe we have helped a number of academic groups and consortia to develop either new methodologies that lend themselves to drug development or even new products themselves. A little more formal is another tool we have that's called scientific advice or protocol assistance. And that is for groups, both companies, but also academic groups who are in the process of developing a product, usually in early stages, and then they come to us and ask what is needed from your perspective as a regulator in order to move this product forward. So what are the types of preclinical studies, animal experiments, what are the quality requirements, and what are the type of clinical studies you wish to see in order to be able to give this a license? What is the information you want to see? So I will not go through all these, the text here. You will be given the slides. It's just to give you an impression that we're open for that kind of early dialogues with drug developers. And I am happy to say that for academic groups, there is a fee waiver that is not in place. For industry, this is reasonably expensive. For academics, there is a fee waiver, so you can see us if you are in the process of developing either a product or a method in the context of drug development. Now, what are we doing for medicines that we deem are of high public health value? And probably to say at the outset, we do not treat all medicines and all applications for marketing authorizations the same. We want to give preference, give more resources, work faster for those medicines that we deem to be priority medicines because they are likely to address a major public health need. And what we do here is we try to speed up the processes. We, as I said, throw more resources at those products in order to enable better and faster dialogue with the developers, again, with a view to having those drugs come out and reach patients faster. Now, here is another important tool, and you will hear, presumably over the next weeks, you will hear more about this thing called conditional marketing authorization. And that is the tool that we have to allow products on the market for which there is a huge, urgent, unmet need, but for which there is less comprehensive data than you would normally hope there to be. So in all likelihood, and I cannot preempt what will come out over the next weeks for the vaccines, but likely the vaccines that we are now assessing will, if all goes well, receive a conditional marketing authorization. And that means this is for serious diseases, and COVID without doubt is a serious or life-threatening disease. There is an emergency situation. It's not an orphan situation. But the conditions or the criteria are that the risk-benefit balance has to be positive. So we need enough data to come to that conclusion. And I'll come to that in a moment. And it is likely that we will see the rest of the data that we need to answer still open questions in the near future, so that the applicant will be capable to conduct those studies. And there is an important sentence here. I don't know whether you can see my cursor here. That is this bullet here, and that is in the legislation. And the legislation says that we should give that kind of conditional authorization in a situation where the benefit to public health of the immediate availability on the market will outweigh the risk inherent in the fact that additional data are still required. In other words, this is a trade-off between earlier access versus completeness of data. And I guess no one would argue that COVID is a situation where immediate availability is of the essence. So So much about the processes that we have in place and who we are. Now let me come to the second part, and Josse in his initial presentation very nicely set the scene for what I want to discuss very briefly now. And I split this again into two parts. And I call the first one COVID for obvious reasons, because this is very topical. And I will try to convince you to believe me that COVID is, in terms of drug development, a very exceptional case that in many ways runs counter to the secular trends in drug developments that we are experiencing right now. So COVID hit us like the odd one out, the complete exception to the rules we are now seeing evolve. Let's start with COVID then. Why is it an exceptional case? And I will come back to this in the last part then. And I will talk about now how we regulate COVID, both treatments, but mostly vaccines. And I will confine myself to the clinical evidence generation. We will not talk about the quality part, the pharmaceutical part, the preclinical. Why is it an exceptional case? Because we have one large global target population. The treatment target is the same for all patients, at least for vaccines and those treatments that directly target the viral infection and replications. So the treatment target, and I will say more of this in a minute, is the virus. And the virus is, if we discount for a moment mutations, the same for all of us. So in a way, you could say one size fits all. Then, at least for the treatments, I'm not talking about the vaccines here, the effect sizes are expected to be small, which has implications for the type of trials we need. And the numbers of patients available for randomized controlled trials, RCT stands for randomized controlled trial, the numbers are high, at least if we look at a global scale and at least during the peak of the pandemic. Here comes a caveat, and Josse has already alluded to that, and we have learned a lesson during the Ebola epidemic. The numbers were high at the beginning, but by the time we wanted to start and test vaccines, treatments, the peak of the pandemic, in that case, the epidemic, was over. And suddenly, these RCTs, or more complex trials, were no longer able to recruit patients. So we had some failed trials. And that was a worry for us. Would we have enough patients to conduct all these trials? And as Josse hinted at that, we were and we still are very worried that some of the small trials that sprang up here and there will not be able to recruit. There will be competition for patients, and we will see, and actually we do see, a lot of failed trials. Now, there is another feature here. Josse, at the end of his presentation, spoke about non-RCT methods. You can borrow evidence for external control groups, and that is something we will come to again very shortly. But that is not a good option for COVID. Why? Because COVID is a moving target. Already over the first months of this pandemic, we have learned to treat patients much better. So the standard of care is improving fast, and you cannot compare then a treated cohort with an external cohort, because there is a thing called drift. In other words, the patients that had been treated a month ago fared less well than the patients that will be treated today, because we are accumulating experiences and learnings. So from all that follows that large, well-planned RCTs, ideally multi-hour platform trials, are doable and seem like the only useful way to generate robust evidence. We have written about that. We have advised the scientific community. we were, shall I say, partly successful. We know there are still small trials coming up, 2-arm, 1 experimental, 1 control arm. We do not believe that this is making efficient use of resources and available clinical trial patients. But we can discuss that later on. So now I want to share with you some thoughts that we have come up with regard to the COVID vaccine approvals. And you know, this is as hot as it gets. Some countries have already issued emergency use authorizations. In Europe, we will likely see that happen in the next few weeks. And these are some of the requirements that we have told the industry. If you develop a vaccine, these are the requirements we want to see. And as I've written here, it wasn't just the EMA, the European Medicines Agency. We did that in collaboration and in harmony with other major drug regulators from US, Canada, Australia, Japan, others. So we have agreed on these key principles so that we can enable one global large vaccine development. At the beginning, there was discussion because of the urgency. Do we really need large-scale so-called phase 3 efficacy trials? And there had been voices saying that, well, what if we show in a couple of 100 healthy volunteers that this product elicits an immune response, antibody titers, T-cell responses? Wouldn't that be enough? And we had agreed early on, the answer is no, for a number of reasons, because that is no guarantee that such a vaccine, even if it produces an antibody or a T-cell response, would be truly protective of the disease itself. So very early on, we took that decision. No corners can be cut. We have to see efficacy trials. And what does efficacy mean? That is written down in the next bullet. That means the endpoint in these pivotal trials is not antibody formation. It's not immunology. It has to be lab-confirmed COVID-19 disease. In other words, it reduces the number of diseases in the treatment population compared to the control group. So treatment group versus control group. Now, there was much discussion on how effective does a vaccine have to be? And let me be clear, we have very few vaccines for any disease that are 100% effective. Some do actually, but many are not. And we don't want to let the perfect be the enemy of the good. So if we had a reasonably effective vaccine that protects most people against the infection, that would be better than nothing. So the question was, well, how effective is effective? And then we agreed that companies should design their studies to be able to show that the vaccine is at least 50% effective. And then what you can read here are some statistical considerations. And we came out with that requirement, and that dictated the size of the trials, which in the end resulted in trials that are now around 30 to 40,000 people. So these are huge trials, but we needed to be able to observe enough cases, and by cases I mean infections, to judge the degree of efficacy. And then we said the trials should attempt, and the word attempt here is important, to assess vaccine efficacy also against severe disease, because that is important. We all know that the severe disease is more important than a mild disease. If you get mild disease, well, it's bad, but the real danger for public health is the severe diseases. So, but we also knew if we prescribe that as an endpoint, the trial sizes would go up to 100,000 or higher. We knew that, and that would severely hamper the development of these products. So that's why we came up with that language. Again, that was trying to do the best in a situation where time is of the essence. Now, what about the safety? We knew that there is skepticism out there in the population. There are anti-vaxxers, there are cautious people who say, well, if the safety isn't sure, I will not be vaccinated. So we knew we needed to have data from several thousand individuals so we can rule out rare, at least reasonably rare adverse events. We cannot rule out very, very uncommon harms. That is, even with a study of 40,000 people, that is probably not possible. And we also knew that we know that from experience, most adverse reactions occur within 4 to 6 weeks after vaccination. And therefore we stipulated that we had to have at least 6 weeks post-vaccination observation periods. Now, you could say that some adverse events may manifest later, and that is true. It is rare, but it is not impossible. So therefore, we also said we want, even if you do the analysis after, say, 6 weeks, we want that trial participants will be followed up within their randomized groups, if possible, for at least 1 year, so we can get a grip on potential late adverse reactions, and also to assess whether there is a waning of protection, because that is also a big question. How long will the protection last? And lastly, we stipulated that while it is good and important to have efficacy studies, and efficacy means how good is the drug in a clinical trial, which is an artificial setting, we also need effectiveness information. And effectiveness means how good is a treatment or a vaccine in real-world conditions of everyday clinical practice. So we will collect those data from vaccination campaigns to better understand long-term protection and long-term safety. So that is a glimpse of what is now going on in COVID. And as I say, all of this is playing out while we are speaking to each other here today. Literally today, next week, the week before Christmas. This is when we will look at that. Now, let's take a big step away from COVID and pretend COVID had not happened. Then we would only be looking at the type of drugs that I'm now going to describe. So these are the secular trends, oncology, other diseases, except COVID, the exact opposite. If we look at the products that we had authorized over the past 2 years, 18 and 19, and you can read this here. This is a mix of chemicals, biologics, already cell gene therapies. And around 40% of these are for very rare disorders. And you see the prevalence criterion written down on the slide here. Now that looks very, very different from the products authorized 20 years earlier. So if you take the year 98, 99, 20 years before, every drug aimed to be a blockbuster for large treatment eligible populations. Most were chemicals. You had a handful of biologicals. You had no cell and gene therapies. What are the projections for near-term future? The trend that I have shown here towards complex biologics, cell and gene products, and for rare diseases will accelerate. And we estimate that in a couple of years' time, we will authorize 10 to 20 cell and gene therapy products each year. That is a major departure from the landscape, say, 20 or 10 years ago, and even today. What are the implications of this? Well, the first one that is very much underappreciated and I think poorly understood is the fact that the nature of variability or heterogeneity is changing. Bear in mind that drug development and drug regulation is haunted by heterogeneity. This would be a piece of cake if every patient reacted in exactly the same way to a given drug. Then it would be easy. The difficulty is that we have heterogeneity, which gives rise to variability. But if you look at the 20th century statin type drugs, I call them statins because statin type because these were the blockbusters of yesteryear for hundreds of thousands of patients. Of course, there were differences in effect size from one patient to the next and across subpopulations, but they were usually explained by differences in downstream pathologies or other between patient differences. For example, comorbidities, patients with renal disease reacted different from patients without renal disease, and so on. But here comes the key point. The interaction between the drug molecule and the drug target in the human body was mostly the same. So if you gave a statin to 100 patients, the statin would interact with largely the same molecule in the human body, which in that case is an enzyme. And now we look at 21st century drugs. Here, the big difference is that heterogeneity even affects the drug-target interaction itself. This was unheard of in the past. I'll give you an example, and the case in point is a disease called cystic fibrosis. We know the cause of cystic fibrosis. It's a monogenetic disease, but there are more than 2,000 known mutations that cause that disease. So if you now develop one molecule, and we have such molecules, that interact directly with a gene product of that gene, that molecule, when administered, say, to 100 different patients, encounters different drug targets. So some of these drug targets are a very good fit for the molecule, but others are a poor fit. And in other patients yet, there isn't even a protein because it is a missense mutation. So if I were a molecule, I would be confused. Now, that is the differences in target. What we are now seeing is also differences in the drug itself. You may be aware that we have authorized cell products called CAR T cells. These are autologous cells taken from patients. That means, and re-administered to that same patient. That means that no two patients will ever receive exactly the same product. Contrast that with simvastatin, for example, or a drug like aspirin. Everybody got exactly the same molecule. We are now seeing that with platform drugs. For example, individualized antisense oligonucleotides. There is at least one disease now, or two already, where you have rare diseases. You have, say, 10 patients with a disease, but they have 10 different mutations. And what you now do is you construct for each individual patient an antisense oligonucleotide. So the backbone, the sugar cover of those drugs is the same for all 10, but the sequence of the nucleic acids is different for each and every patient. Now, are you looking at different drugs here or the same? And we will not only see that for antisense oligos, we will see exactly that for other so-called platform drugs, for fusion proteins, for antibody fragments, antibody drug conjugates, interfering RNAs, cells, as I already said, and even personalized treatment combinations. So different patients receive different drugs, and a given drug will see different targets. That has now major implications for evidence generation. The obvious one is we now have smaller target populations. Remember the statins, there was talk, and that was only half joking, should we add simvastatin to drinking water? Because pretty much everybody under the sun is or will become target patients for that drug. And it was only just an exaggeration, as I say. Now we go in the exact opposite direction, smaller and smaller target populations. And at the extreme, remember the example that I gave about the antisense oligo? At the extreme, the target population for a given molecule equals one patient. The consequence of that is extrapolation of efficacy information from one subpopulation to the next is now totally different from a statin type situation. In statins, you could say, well, if it works for secondary prevention, it may also work for primary prevention with different adjusted efficacy estimates. Well, maybe that's true. This is no longer possible in a situation like the cystic fibrosis, where you have different gene products, different targets. Our attention on what is a drug and what is a disease will have to become far more fluid. Remember the antisense example. So we have 10 patients who get 10 different antisense oligos. Are we looking at one archetypal drug with variations? Are we looking at a drug class? Or are we looking at a multitude of different individualized drugs? And the implications for drug regulation, drug development are of course huge. And to make matters even more difficult, some drugs' properties may shift over time. And that was unheard of. The formula for aspirin was devised over 100 years ago, and aspirin is still the same molecules today as it was 100 years ago. But now take the CAR T-cell preparations. These are very complex preparations. And we are learning over time how to prepare these cells better and better and better. So, are the products that we authorized 2 or 3 years ago still the same products that are being dispensed today? And you could argue that because we have now better manufacturing processes, we now have different biological properties. So are we looking at the same product or are we looking at a new product? Are the clinical trials that were the basis for marketing authorization 2, 3 years ago still relevant for those new products? And this is not just the CAR T cells. We have authorized a gene product, and almost at the time when we authorized it, the company discovered that they could produce this in a much better way. And why should we not allow a better product on the market? But now all these studies that we have seen, are they still relevant for the new product? So what you do is now to have bridging studies, etc. It is becoming a lot more complex. So clinical evidence generation for complex 21st century drugs will require different tools and study types than what we had for the good old aspirin or simvastatin type drugs. Now, something else has changed, and that is the number of research questions that are now being asked by stakeholders. Patients say, well, I'm not interested in endpoint 1, I want to see endpoint 2. I'm interested in quality of life or in relapse rates or whatever. And I call that the depth of information, and you can see that in the middle of that graph here. So different patients want to see different endpoints. But there is also something what I call breadth of information, and that is different patient subgroups. And I have put this down here. This is depicting the research questions of a hypothetical drug that is not a real drug. But on the one hand, we have different endpoints, long-term efficacy, rare adverse events, the depth. But then we have the breadth of information. We have mutation 1, mutation 2, mutation 3. We have early stage disease, late stage disease, etc. So we have different subgroups of patients. And that is not enough. We now have people who are asking questions about the context of information, and that is on the vertical axis. And these are usually payers or health technology assessment bodies who say, well, it's all well if you compare that new drug with comparator 1, but we are using comparator 2. So how does your new drug stack up against comparator 2? And another payer may say, well, in our healthcare system, we will combine this new drug with drug X. How does that fare? And someone else says, no, no, it's combined with drug Y. So the result of this is we have this huge complex matrix of questions that are all relevant to some stakeholder, but that are impossible to answer. And what that slide also means to show is that you will only be able to show a very, very small fraction of all these research questions by way of randomized methods. And whether that is your classic two-arm RCT, or whether that is one of the more complex designs that Josse has just alluded to, sorry, the world is too complex. We cannot answer all these questions by way of clinical trials, interventional clinical trials. And what I try to depict here is we will have to take recourse to other types of knowledge generations. So for example, the greenish boxes mean that you do indirect comparisons, or you take an external control arm. So you're borrowing information from real-world data, from other clinical trials. In some cases, you will only be doing something that is usually a no-no. That's called before-after comparisons. And all the statisticians will throw up their hands and say, well, that is prone to bias. That is not robust evidence. True, maybe, but we cannot do better. And the ugly part of that slide, and that is a very unwelcome message, is those light gray cubes or bluish cubes. And with that, I mean to show that a large number of research questions will never be answered because we cannot answer them. We have practical constraints. Now, to make matters even worse, let me introduce you to this matrix. I have already said.
Sorry, Hans-Georg.
Hello?
Yeah, we need to try to finish, to leave a little bit of time for discussion. Yeah. If you can finish in 2 minutes.
Thank you. I will finish. Yeah.
Thanks.
Okay, I'll speed up. So what we have is small target populations is the future. We had large target populations in the past. But we also have drugs, a few with large effect sizes, very good. But we also have a lot of drugs with small effect sizes that may still be important and valuable in the eyes of patients. How do we generate evidence? Well, let's start with this quadrant. You have a large target population and a large effect size. Very easy to show by randomized control trials or even by non-randomized methods. I call it real-world evidence, observational studies. No problem here. Let's go to this. You have small target populations and large effect size. You can perhaps even do a small RCT because large effect sizes can be demonstrated in small trials. But you can certainly do observational studies and they can be credible. What about here? You have a large target population and a small effect size. That is the classic statin situation. This is where you do a large RCT. All fine so far, but the future is small target populations with smaller effect size. And what will you do here? You cannot do an adequately powered RCT because you don't have enough patients. You could perhaps do observational studies, but because they're prone to bias, they are not credible or poorly credible. So now we are ending up with something that again is unwelcome, unavoidable uncertainty. So what are the consequences for drug development, regulation, and pricing decisions? The inevitable future is a combination of randomized and non-randomized methods. We will have to draw on a variety of data sources, with data being prospectively and retrospectively collected, and we will need to follow up patients for prolonged periods of time, including post-authorization. My last slide. The rise of these new products, personalized medicines, will require from all stakeholders the acceptance of the notion of unavoidable uncertainty. And I know that doesn't sit well with many of my colleagues. It will require transparent communications and also communication on uncertainty. It will require fit for purpose, additional data sources and additional methods, analytic methods for non-RCT evidence. Better methods for evidence synthesis from many types of clinical studies and trials. We will need rigorous on-market follow-up of clinical outcomes in the majority of patients. And we will require a regulatory and payers framework that allows frequent assessment and refinement of decision as new knowledge accumulates. We call that the life cycle approach to drug regulation or payers. With that, I thank you very much for your attention, and I hope there is sufficient time for discussion. Luciano, over to you.
Thank you very much, Hans-Georg. Really an excellent presentation. I really appreciated also your remarks and your presentation about the very modern aspects of drug development. Your reference to CAR T cells, to gene products, I think that is extremely interesting for students and young researchers, young graduates online, because some of these topics, very often are not treated in depth in the curricula at the university. So I think that's a very valuable information. So indeed, we are a little bit late, but I think it was worthwhile to have really these two excellent presentations by Professor Thomas and Professor Eichler. So now I think we will take a little bit more time than the 2 hours that we planned. So I apologize in advance for the participants, but I ask you to stay online 10 minutes more because also this part of discussion is very important for us. And I would like to ask the three discussants, Maeva, Chris, and Sibel, please just address one question each to the two speakers, and then we will ask them to reply to your questions, and then we will have a final discussion with some questions coming from the chat. So please, Maeva, the floor is yours.
Thank you, Luciano. And I will also like to thank very much our speakers, Mr. Josse and Mr. Hans-Georg, for their presentations. It was very interesting and full of information. I will just, try to put a quick question to leave time for the questions in the chat as well. You mentioned the importance of the post-authorization follow-up. I was wondering if there are the necessary procedures in place are able to ensure that this is the case, and if any actions can be taken whenever there are any unexpected situations that come up during that follow-up period. Thank you.
Thank you very much, Maeva. We continue with Chris, Professor Schravendijk, please.
Yes, I was also very much pleased with the two talks. I really enjoyed all the information, and my question to both speakers would be that in the COVID pandemic crisis that we have all seen happening, there was a lot of panic in the springtime in the emergency hospital floors and care centers about how to help the patients. And suddenly during the summer, in all the confusion, there was suddenly the dexamethasone. And it was suddenly quite clear that this was something that could really help the patients. So for both speakers, I would ask, how did this happen? Because dexamethasone discovery was a real success. Was it done by an RCT? Was it done by maybe better communication between doctors and healthcare workers? How did it happen? Because it happened actually quite fast. Suddenly we had dexamethasone, and the outcomes possibilities of all the patients in the world became much better in a short period, and still are much better than in March. So this is my question. Is it really only the RCTs or other more complex trials, or was it better communication between primary healthcare workers on the work floor itself, worldwide, I mean?
Thank you very much, Chris, for the very good question. And then Sibel, please.
Thank you for all the speakers, and thank you, Maeva, the EPSA representative, and thank you, the UNICA president, Luciano Saso. My question is easier than Chris's question. It is more complicated and the answer will be long probably, but my question is related with the designs that Professor Thomas mentioned. The novel clinical trials, and we have seen many designs, and we know many clinical trials are on. So how you decide on which clinical trial is suitable, which design? Because it is very important. Maybe this answer is will be very informative for the young professionals who are listening to us. So decision on the designs, which one to choose for the trials, do you have any algorithms or any program to use? Which one is the best? Thank you.
Thank you, Sibel. So now the floor over to the two speakers to reply to these three questions. Who would like to start? Maybe just in order of
yeah, okay, I can start yes. With some of it. The first one was about post-marketing authorization safety follow-up. Perhaps more a question for Hans-Georg, but I will certainly try already to give a number of elements. With conditional approvals, as they are probably going to be the case for the vaccines, but it is already a type of marketing authorization that was used since a number of years. There is the commitment of the marketing authorization holder to do a number of extra studies post marketing authorization. And for safety, they are known as post-authorization safety studies, PAS studies. That is something as a commitment, and there are studies that are specific on the safety, long-term safety in the real world, not within clinical trials. It's still a clinical trial, but more pragmatic. So the inclusions are different, of patients are different. And the second thing here, I think that for the COVID vaccines, it would be interesting, and I don't know whether it will be imposed, to create a register or several registers of the vaccinated people and follow them up for a number of months, eventually also a year or longer, as an observational type of trial. That is already some elements that I could add to that. I will leave Hans-Georg to answer his first question, the first question.
Okay, thank you. Yes, no, I agree with everything Josse said. Just to say, the commitment is not just for conditional marketing authorization. No, every new drug now has a risk management plan.
Yeah.
And that can have, as you just say, a post-authorization safety study, but there is something else also, post-authorization efficacy study. If we want to know more about the efficacy, we can also impose that. So yes, that is in place. Yes, that is enforced. We have analyzed and that is published. Do the companies live up to these promises, to these agreements that we make with them? And the answer is by and large yes. Sometimes the information comes in delayed, but it does come in. So this is being done, and I think this is a success, this life cycle approach. Now, Josse said about the COVID registries. Absolutely, that will be the only way to monitor and survey the long-term efficacy and also long-term protective effect of these vaccines. And yes, we have in place a number of registries. The EMA has entered into contracts with a number of academic institutions to enable us to get access to such data. So we will sort of online analyze these data, and as information rolls in, we can update the benefit risk. Over.
Yeah.
There was a second
If
question.
I can have a go for the second question about the dexamethasone case within the COVID pandemic. Why was it so successful and so quick? I think there are a number of things to say about that. There were two platform trials going on. The one from the WHO, the Solidarity trial, where dexamethasone was studied, and the Oxford trial. So I think the fact that they were initiated as platform trials with different drugs gave us a good possibility of comparison with sufficient number of patients. And it is also because, as Hans-Georg already said, because the pandemic was still very lively ongoing, the number of patients that you could include quickly and fast was important. So that reduced the time you needed for the study, because the number of the recruitment was very quick. And then finally, it was a collaborative effort. And when you collaborate with different stakeholders, I think it's easier to come to an answer.
Thank you, Josse. And Hans-Georg, maybe on the same topic.
Yeah, no, totally agree. The only thing I could add is, don't forget, Chris asked, why is that happening so fast? Sorry, I should switch that on again. Why did it happen so fast? Well, don't forget one thing as well. These were not new drugs. All of these drugs were what we call repurposed. We have had dexamethasone on the market for ages, longer than I can remember. And the other drugs that were studied here were also repurposed. So, and then some smart brains thought, well, this is the mode of action. We know the safety profile. We know the drug. Maybe that will help. Sadly, actually, dexamethasone, as it stands now, was the only one for which it panned out. Some of the others, notably hydroxychloroquine, and you know the political story behind that, did not pan out so very well, and others as well. So we had to ditch those. So as Josse said, these large multi-arm trials are ideal when you have an urgency and when you have a lot of repurposed drug. Then you can move very fast. Thank you.
Thank you. And then the last topic, the question by Professor Suzen.
Yeah, a more difficult one. What is the best practice, and is there an algorithm to choose what type of clinical trial design you need? No, I'm not aware of that. It is a case-by-case thinking about the strategy that you need. There are a number of guidelines, of course, and guidance documents for these types of adaptive designs and complex trials, but whether you are able to use them within your development program and how you will be able to use them is up to the sponsor in dialogue with the regulators during the scientific advice.
Thank you. And Hans-Georg?
Yes, But don't forget, very often, you don't have the luxury to say, oh, shall we do A or shall we do B or C? Sometimes you have to make do with what you have. And I spoke to that, for example, with those rare diseases or with the gene therapies. You cannot do any randomized control trial because there are so few patients, or because there is ethical caveats. Patients will not want to be randomized. But when it comes to these complex trials, the multi-arm, there is a practical barrier. So we now looked at the situation where this was eminently doable. COVID came, we had 5 or 10, whatever, extant means they are already in existence. Drugs that are here, that are cheap, that are available. Now you can do this multi-arm trial. But now think of a different situation. And we've tried that in pediatrics, for example. Pediatrics is a situation where companies compete for patients. You have only a small number of patients. Excuse me. You have several compounds. Why could you not do a multi-arm trial? And as Josse said, you save patients, etc. Well, that's all very good. But one product is already in late stage. Another product will only come in 2 years. So what should you do? The first is ready now. The second isn't ready. So we tried that, but you very quickly stumble into practical difficulties unless you are in a situation where you have many, many candidates at the same time. So such a situation is, for example, breast cancer, colon cancer. These are the rich indications with a full pipeline. Once you step outside that rich pipeline, you will soon reach the limits of what is feasible. And then you are happy if you can even do a 2-arm RCT. Thank you.
Thank you very much. So it's a little bit late. Unfortunately, there were some other questions in the chat, but there is no time to answer them. I apologize for that. But I would like to launch one final, big topic. I know that is quite big and difficult to cover, but I mean, I would like really to have a comment from the two speakers about the role of artificial intelligence and big data in drug development in the near future. Also because this, I think, has a practical impact on the curricula of pharmacy and biotechnologies and other degrees, in Europe and in other regions of the world. So how much we should actually teach today about these topics to make sure that our students, when they will graduate, they will be ready for this new world. Because of course, Professor Eichler mentioned the smart minds. If we think of the smallpox, we have to remember Edward Jenner or Pasteur or other people who discovered drugs really only with their ideas and knowledge from themselves. Nowadays, I think thanks to, again, artificial intelligence, etc., I think we can spot some interesting phenomena related to drugs, allowing us to maybe speed up the discovery of some drugs. So can you please elaborate a little bit on this big and large and complex topic?
Well, I think that there is potential, of course, for these technologies, but they are not yet widely used. I think artificial intelligence is already introduced in drug design, less in drug development, and surely less in clinical trials, but there are already initiatives of virtual clinical trials, and if there are certainly things that can be done in the future. And for big data, that is something that surely can be developed for real world evidence, where there is a lot more data available than just the data of clinical trials. So certainly possibility for developments in the future.
Thank you.
Yeah, so if you ask me, should this be taught to students today, I would say absolutely yes.
Yeah.
I think there is very little doubt that, don't forget, someone who's a student today will only become a professional in 10, 20 years, sort of at the peak of their career. So at that time, very, very likely, yes, there will be wide use in that field for AI. Whether it will happen in the near term is a different matter. And I agree with what has been said. There is very little now in clinical development. There is more in the drug discovery field, etc. I can give you an example where we foresee that it will happen very soon in regulation, and that is in signal detection. We get a lot of reports from physicians who think that reaction A has been caused by drug X. Now, is that really a signal or is that noise? And we have been struggling. There are formulas, there are algorithms. So we will possibly, and I have to be very careful, see very soon that these processes that are always going on in the back as we speak, we do this on a weekly basis, will be augmented by AI probably in very few years. That is where I see inroads coming soon.
We lost you, Hans-Georg. Unfortunately, we lost you. So maybe, yeah, are you? We lost you for a moment. Are you?
Yeah, sorry, I was off, but
okay.
I was already done. So that was basically my message. I'll stop here.
Okay, thank you very much. So again, I think it's really very late. Let me just mention, maybe we have time for a very short again, question from the chat. We try to cover this one also. The question was, how do you ensure that companies enter their investigational drugs in umbrella and platform trials? Could the potential for between product comparisons scare them off due to the competition with other companies? So can you please comment a little bit on this question?
Yeah, there is certainly a friction between, what you would like to see, that is collaborative trials, and what you see now, more competitive trials. The industry is not very keen to do comparative trials and multi-company trials. But that is very interesting. That would be very interesting to do, and would generate evidence that is more robust than that we have today. And I don't know whether regulators could make that an enforcement. That would be ideal, but I don't know whether we are close to that, yes or no.
Thank you. And Hans-Georg?
Well, the answer to your last question is, well, we're not very close to it. And again, the reason why this was successful in COVID, because these were repurposed drugs. These were not de novo developments. And if you watch what is happening in the COVID field, there are now treatments on the way, and these are new treatments. And the companies who developed those did not wish to be part of the multi-arm trials that were sponsored in the US by the NIH or by others in the UK. No, they wanted to go alone, and they're doing this now. So we will see de novo developments reasonably soon. No, we cannot force them. So I think that's all I have to add.
Perfect, thank you very much. There is another interesting question from Katou Mouy from the Faculty of Medicine and Pharmacy of the Free University of Brussels, but we will kindly ask Professor Thomas maybe to reply by email, because really now there is no time to address this additional question. But again, I would like to thank very much the two excellent speakers we had today, the three panelists and discussants. Again, I would like to thank very much the partnership of EPSA, the European Pharmaceutical Students, and EMA, the European Medicine Agency, for organizing together this event. And let me thank also all the participants for being online in this afternoon with a good percentage. I mean, there is of course a dropout in the last few minutes, but this I think is physiological, but I mean, the participation was quite good. Let me thank also the UNICA office, Laura Colò, who organized this event so well. So see you soon online for other UNICA student webinars like this one. Very soon on the 14th of December, we will have another webinar on career development in the pharma and biotech field. So I hope to see some of you online again. And again, thanks a lot to all of you, to the speakers. Thanks. Bye-bye.
Bye.
Okay.
Thanks.